Steps in cloning with Lambda. So different steps can be used for cloning with Lambda, and of course one of the first step is that कि जो DNA है Lambda genome, इसको फेज से आइसोलेट किया जाता है और इसको कट किया जाता है किसी मुनासिब रिस्ट्रिक्शन एंजाइम से. And after cutting the Lambda genome, so what will happen? We have the idea that 3 fragments may be produced. 2 fragments that are present at the right or left side of the Lambda genome, they are essential because they encode vital proteins for the survivability of the Lambda virus. Whereas the central region, that is a 3rd fragment that will be produced, it is non-essential. So it is removed, and in the 2nd step, the foreign DNA, it is ligated with 2 essential fragments of the Lambda genome, so that now we can get a recombinant DNA that contain 2 fragments from the Lambda genome, the fragments that is present at the right or the left side, and the filler DNA, it is replaced with foreign DNA. And here the length of the recombinant DNA molecule that is produced, it is very, very important, because if 2 small fragments are produced that is smaller than 40,000 base pairs, they will not be packaged, and even larger than 53,000 base pairs, they will also be you can say that removed or will not be packaged within the phage head. So this is the constraints that we are going to use as a selectable marker in recombinant DNA process. So only foreign DNA that is up to the size of 53,000 base pairs, they can be packaged. Very larger and smaller DNA fragments, they are discarded. And in the 3rd step, if we have produced the recombinant DNA molecule, and in the 3rd step, it is the in vitro packaging. In vitro packaging means that we are mixing the recombinant DNA with essential viral proteins. So here we can use the crude extract of the bacterium, जो कि सारी की सारी जो मुनासिब रिक्वायरमेंट हैं या ज़रूरी रिक्वायरमेंट हैं वो प्रोवाइड करेगा, जिसमें पैकेजिंग प्रोटीन्स हैं जैसा कि हेड और टेल प्रोटीन्स. तो इन विट्रो पैकेजिंग होगी, और इन विट्रो पैकेजिंग के बाद जो 4th स्टेप है वो है ट्रांसफॉर्मेशन ऑफ इकोलाई. Transformation of E. coli, it is very efficient when we compare it with plasmid vectors, where the transformation efficiency may be a little bit low, but when the phage particles, they are used as a vector, the transformation efficiency, it is very high if we are able to produce viable phage particles after recombination. When infection is there, then of course after some time the phage particles will cause the lysis of the host cells, so plaques are produced, and the plaques they are selected for the recombinant phages. When recombinant phages we isolated after lysis, तो जो लास्ट स्टेप है या 5th स्टेप है वो हम ये देखना चाहेंगे कि क्या हमारा फॉरेन डीएनए इसमें स्टिल प्रेजेंट है कि नहीं. तो उसके लिए हम डिफरेंट टेक्निक्स जो हैं वो यूज़ कर सकते हैं. जैसे स्टार्ट में आपने देखा था कि we can perform hybridization studies on the membranes where the foreign DNA, it can be immobilized on the membrane, and by using a known probe we can detect our gene of interest. Another technique is that we can use DNA sequencing to detect our cloned fragment, and other techniques like some selectable traits, they can also be used that we are going to discuss after some time. This diagram, it indicates the steps that are involved in the cloning procedure by using bacteriophage lambda vectors. As I told you that the DNA that is present between the left or right side, it is non-essential and it can be removed. And after removing or digestion with restriction endonucleases, this fragment it can be replaced with the foreign DNA here between the left or right region, so that we can produce a recombinant DNA molecule that contain the two essential fragments of the phage lambda, like one at the left or the right side, and third one it is the foreign DNA. When we produced the recombinant hybrid DNA, the next step is that the foreign DNA it can be transformed into the phage head by a process which is called as the in vitro packaging. So it may be inserted into the phage head and assembly process is there, and now this virion, the phage particle, it is now ready to infect new host organism. So these are a few steps that are involved in cloning with bacteriophage lambda. Now we have the idea that modified lambda phages that are chiron 41 or chiron 16, they contain reporter genes, as I told you, that beta galactosidase, and if the foreign DNA it is inserted, its expression it will be lost. That will indicate the presence of foreign DNA. That we are going to discuss after one or two topics, that how on the basis of beta galactosidase we will select our recombinant DNA.